Okay, so chapter 10, it's about muscle histology and physiology. So in this chapter, as I told you before, we're going to learn the characteristics of the muscle types that we have. We're going to compare and contrast between smooth, skeletal, and cardiac. And we're going to see the mechanism of contraction and the components of muscle cells like sarcoplasmic reticulum, sarcolemma, what is a triad, what is a sarcomere. So we're going to look at the ultrastructure of the muscles. And we are going to see muscle contraction, relaxation, some molecules that play a role in this process. And we're going to see the metabolism of the different muscle fibers. So why is it important to learn about muscles? Well, because muscles are important for us to move and to produce force that we can carry objects. And then it helps us as well to maintain posture and generate heat. Whenever you have been in a long period of fast and you are not still close to get your next meal, your muscles start contracting to generate heat so that you don't feel so cold. And then we also use contraction of the muscles to propel blood, to help to transfer items like food from these different parts of the digestive system into others, and also to release undigested food. So we have sphincters that controls openings. So muscles then perform this function of contraction and relaxation in which they convert chemical energy into mechanical energies for our body to function. So the types of muscle that we have skeletal, cardiac, and smooth. Skeletal, it is found attached to the bones for the most part, and the skin in the case of our muscles of facial expression, if you remember. And then cardiac muscle is located only in the heart, and smooth muscle is located in hollow organs like the uterus, fallopian tubes, the intestines, etc. In terms of shape of the cells in the skeletal muscle are long and cylindrical. They don't branch. And in the case of cardiac muscle, they are short, cylindrical, and they branch. And in smooth muscle, they have a thick middle part and taper ends, so it looks like a spindle. Now, why these two are called striated, skeletal and cardiac, while this one is called smooth? Well, because those striations are bands that runs across the short axis of the cell, forming a pattern that looks like striations. And then in the smooth, you don't have that. And then the two ones that are under involuntary control will be cardiac and smooth muscle. They are controlled by the nervous system. While this one, skeletal muscle, is under voluntary control. And then in terms of function of cardiac muscle will be to pump blood continuously. A smooth muscle moves substances through organs, or allowing the exiting of substances outside of the body. So common properties of all muscle type, it is contractibility, so ability to shorten its length forcibly, excitability, responding to a stimuli, conductivity will be to transmit electrical signals, distensibility, being able to stretch without damage, and then elasticity, to return to the original shape and length after being stretched. So here are the characteristics of skeletal muscle. You already seen this before. This is one cell, another cell, another cell. So 3 cells together. They are long, cylindrical. Another name for muscle cells is called muscle fibers. So these cells, they form from a fusion of cells, precursor cells that are called satellite cells. And when satellite cells fuse, many of them, they form a single skeletal muscle cell that end up with multiple nucleus. So these will be the dots that represents the nucleus. So, and then you have these bands across these cells. Cardiac, cylindrical but short. So they connect these individual cells and they only have one nucleus per cell. So this is uninucleated, the other one was multinucleated. So this is one cell connected to another one, connected to another one, this one. So these branches. It also has striations, and in between the striations, at the end of these cells, you have these intercalated discs that are specialized connections. So these intercalated discs are like serrated and helps for these muscle cells, the cardiac muscle cells, to connect so strongly because these are the ones that contract much stronger than the rest and prevent separation of these fibers when you are propelling blood. Thick in the middle, taper on the ends like a spindle without striations and only one nucleus, and short fibers will be smooth muscle cells. You already saw part of the membranes that covers the muscles. And we said that the muscle, it is made by 10 to 100 cells together forming fascicles, and several fascicles form a muscle. So one muscle has several fascicles, and the fascicles have these muscle fibers or muscle cells, 10 to 100 per fascicle. And then inside these cells, we have myofibrils. And these myofibrils are made out of proteins that are called myofilaments. So then, this is the organization of muscle. Muscle is made of fascicles, fascicles are made of muscle fibers are made of myofibrils, myofibrils are made of myofilaments. And then we have, again, this endomysium that surrounds individual muscle fibers. And then the fascicles are surrounded by the perimysium, while the entire muscle is surrounded by an epimysium. And then you compartmentalize these muscles, and they have a specific function. Now, inside the cells, these muscle cells, we have different structures, and they have a specific name. Now, we have a plasma membrane that surrounds these cells, which is called the sarcolemma. And then we have the sarcolemma that extends deeply into the cytoplasm, forming a tubule. This, you call it a T-tubule. And then on the sides of the T-tubule, you have the calcium storage organelle, which is the sarcoplasmic reticulum. And then we have inside the cytoplasm of these cells, we have the myofilaments, thick and thin filaments. So these are components of the muscle cells. Sarco means flesh in Greek. So it's the same organelles, but you call it sarco in the case of the muscle fibers. And then the sarcomere is a structure that runs between two bands that are called the C discs, and they form the structure and functional unit of the muscle. So we will see details of the sarcomere. So if this is the muscle levels of organization. So you have a fascia that covers the epimysium in this muscle, and then the epimysium covers the entire muscle that is formed by several fascicles. How many fascicles do you see there? This is one fascicle, an example of one fascicle. Count them and tell me how many do you see?
There's 10.
So this is one.
12.
12, right? So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. If I counted that tiny one. But it's 12, 13. So now inside these fascicles, we have the muscle fibers. And we're not going to count them, but we have many. From 10 to 100 of these forms a fascicle. Now, if you extend one of these muscle fibers are covered by this endomysium, and inside this endomysium, we have the sarcolemma. So the endomysium is not the same as the sarcolemma. The endomysium is connective tissue, like elastic fibers, collagen fibers. It's like a wrap. And the sarcolemma is the plasma membrane. The plasma membrane is made of phospholipids, and it has channels so that the ions can come in and out. And then inside this muscle fiber, we have these T-tubules that are extensions of the sarcolemma, and these organelles that stores calcium, which will be the sarcoplasmic reticulum. And these are the myofibrils. So the myofibrils are collections of proteins. We have two proteins. We have a thick protein, which is myosin, and a thin protein that is called actin, plus other proteins that holds these into their corresponding area, which is titin, for instance. This is an elastic protein that anchors this myosin to this band here and this band in here. So this is what we call the A band or dark band, and this is what we call the light band or I band. And in the middle of these bands, we have the C discs. So in between the two C discs, we have a sarcomere, which is a structural and functional unit of muscle. So we have A band in the middle of the sarcomere and two I bands. And this is the magnification of these proteins. So this is myosin, which has a head, has a neck, a hinge, and has this rod portion. And then here we have actin. And then here we have titin, which is like if it was a spring that it stretches and then recoils every time that these fibers slide over each other. So let's look at the more detailed part of the muscle cells. So the function is contraction. That's why they're long. And then they can shorten during contraction. So again, the plasma membrane is known as sarcolemma. And this sarcolemma allows the entry and exit of ions. So controls ion exchange. We have channels for that. Inside the sarcolemma, we have the cytoplasm, that in the case of muscle fibers, we call it sarcoplasm. In the cytoplasm, we have glycogen. Do you remember what is glycogen? Glycogen is made out of what? glucose. So it's like the bank for the muscle to contract. This glycogen is exclusively used by these muscle cells. If your blood glucose levels drop, the muscle cells will still have been able to contract because they have their own glycogen. We also store glycogen in the liver, but the liver gives the glycogen so that the blood glucose levels increase. But the muscle cells, they use their own glycogen. They have their own savings. And then they have this protein that is called myoglobin that is for energy storage and oxygen binding. And depending on the levels of the myoglobin is going to be whether you have high oxygenation or low oxygenation. And then we have these contractor proteins, which are the myofibrils, which are actin and myosin, that are located in parallel. And then we have the sarcoplasmic reticulum, which is a smooth ER that stores and releases calcium to allow the contraction to happen. And then we have numerous mitochondria because we have to have a lot of contraction. And then we have multiple nuclei per cell, and they are located in the periphery. Why? Well, because you have to have the middle for allowing the contraction of the actin and the myosin. So, sarcolemma, and then sarcoplasm. Inside the sarcoplasm, you have myofibrils with actin and myosin. You have multiple mitochondria, and each of these is surrounded by the sarcoplasmic reticulum, and then you have on the side the nucleus. That's why the nucleus is on one side, because you have to have this in the middle. And this is another view of these cells. And if you compare this cell, muscle fiber, to this red blood cell, see how big is this? This is short. They have different function. This is to go into the tiny blood vessels and go and transport oxygen and then pick up CO2 and deliver all those. While this one is to shorten and then extend so that you can have movement. Where in our body we will have the longest muscle fibers?
In our leg.
Yeah, in our thigh. That's where we will have our longest muscle fibers. Now the sarcolemma extends deeply into this muscle cell in the cytoplasm, and it forms this T-tubule, transverse tubule. And on the sides of the transverse tubule, so if you magnify this part, you have this widening of this sarcoplasmic reticulum. This widening of the sarcoplasmic reticulum is called the terminal cisternae, so that you can have an electrical signal going from the surface of the cell deep into the cell, and this electrical signal will open channels that we have here in the terminal cisternae, channels for calcium. So calcium can exit, and then that calcium is going to make these myofibrils to slide over each other during contraction of the muscle fibers. So this is called a triad, a single T-tubule and two terminal cisternae to the sides, triad for 3. For the myofibril structure, if you magnify one of these myofibrils, again, you can see that you have this A band and these I bands. This region is thicker than this. So when you shine light in the microscope, this dark band appears dark because the light shines less than in this less dense region in the I band. So this is the collection of myosin fibers, and this is actin. Myosin has these tails in the middle, and then it has the heads on the sides, several heads. The thickness of all of these thick filament, it is 50 nanometers. And now, these tails are basically made by a helix of this protein, myosin. And then they do this, like if it was DNA, and then here at the hinge, you have this connection of the tail to the neck region, so that the heads, you have two heads, the heads can move toward the center. So it will be like doing this. Like if this will be the tails of the myosin, and this will be the neck, and this will be the head, you will be doing this. So that connects, the heads connects to this actin, which it has only 8 nanometers in diameter, so it's almost a half in thickness as this. And then you can pull the actin toward the center of the dark band or the A band, shortening the length, so you bring closer the two C bands. So the I band, when you have contraction, disappears, and you have A band. For actin, you have also a helix made of these subunits, which is globular actin. When several of these actins form a filament, you call this F-actin. So you have two F-actins forming a helix, being bound together by this protein that is called tropomyosin. And then this tropomyosin, it is attached temporarily into actin by the aid of this protein that is called troponin. And these actin subunits, they have a site that is called actin active site, in which you will have interaction of the head of the myosin with the actin active site when this one is available. It's not always available because it is covered by this tropomyosin. So you need to have calcium, and this calcium binds into troponin. Troponin let go tropomyosin, and then tropomyosin moves away and then exposes these actin active sites so that the head can connect into here, and then you have interaction between actin and myosin, pulling actin towards the middle, shortening the sarcomere. And then once you have contraction ending, then you have release of these actin heads, myosin heads, and then it releases this bridge with actin, and then titin is going to contract, helping, or is going to pull myosin back into its space so that you can regain the normal length of the sarcomere. So we will end up here, and then we will continue.